The reproductive strategy of certain deep-sea anglerfish species, particularly those in the suborder Ceratioidae, is one of the most extreme and fascinating examples of sexual parasitism in the animal kingdom. This process, known as sexual parasitism, involves the male anglerfish fusing to the female, becoming a permanent parasitic appendage that provides sperm while losing most of his own organs. Below is a detailed explanation of this phenomenon, covering the biological, ecological, and evolutionary aspects. Overview of anglerfish and sexual dimorphism. Anglerfish in the Ceratioidae suborder inhabit the deep sea, an environment characterized by extreme darkness, high pressure, and scarce resources. These conditions have driven the evolution of extreme sexual dimorphism, where males and females differ drastically in size, appearance, and behavior. Male anglerfish. Females are significantly larger, often growing to sizes ranging from 20 centimeters to over a meter, depending on the species. They are equipped with a bioluminescent lure, the esca, which they use to attract prey in the dark depths. Females are the dominant reproductive partners, capable of surviving independently and producing eggs. Male anglerfish. Males are much smaller, often only a few centimeters long, and lack the lure and robust body of females. Their sole purpose is reproduction, and their physiology is adapted to locate and attach to a female in the vast, sparsely populated deep sea. This extreme dimorphism sets the stage for the unique reproductive strategy of sexual parasitism, the process of male-female fusion. The fusion of male and female anglerfish is a remarkable adaptation to the challenges of the deep-sea environment, where finding a mate is difficult due to low population densities and limited visibility. The process can be broken down into several key stages. 1. Mate location challenges in the deep sea. In the deep ocean, where light is absent and individuals are widely dispersed, finding a mate is a significant challenge. Male anglerfish have evolved highly specialized sensory adaptations to overcome this. Olfactory and visual cues. Males have the larger sensitive eyes to detect the bioluminescent glow of a female's esca, as well as highly developed olfactory organs to sense pheromones released by females. These adaptations allow males to locate females over long distances in the dark. Small size and mobility. The small size of males reduces their energy requirements, allowing them to swim efficiently in search of a female. Their streamlined bodies and reduced digestive systems prioritize mobility and mate detection over independent survival. 2. Attachment. Initial contact. Once a male locates a female, he uses his specialized jaws and teeth, often described as pincer-like, to latch onto her body, typically on her belly, side, or near her reproductive organs. The exact location varies by species. Enzymatic fusion. After attachment, the male releases enzymes that dissolve the skin and tissues at the point of contact between his mouth and the female's body. This initiates the fusion process, allowing the male's tissues to merge with the female's. Species-specific mechanisms. In some species, such as Ceratus caruncula, the fusion is permanent, while in others it may be temporary or less complete. The degree of fusion depends on the species and environmental pressures. 3. Tissue integration and parasitism circulatory connection. As the tissues of the male and female merge, their circulatory systems become interconnected. This male's blood vessels fuse with the female's, allowing him to receive nutrients and oxygen directly from her bloodstream. This marks the transition to a fully parasitic relationship. Organ degeneration. Once fused, the male's body undergoes significant degeneration. His eyes, fins, and most internal organs, except the testes, atrophy and disappear, as they are no longer needed for independent survival. The male becomes a streamlined appendage, often resembling a small growth or bump on the female's body. Sperm production. The male's testes remain functional and, in some cases, grow larger relative to his reduced body size. He becomes a permanent sperm-producing organ, releasing sperm on demand when the female is ready to reproduce. 4. Reproductive role sperm delivery. The fused male provides sperm directly to the female, either continuously or in response to hormonal cues when she releases eggs. This ensures that the female has a constant supply of sperm, eliminating the need to find a new mate for each reproductive cycle. Multiple males. In some species, a single female may host multiple parasitic males, increasing her reproductive potential by ensuring a diverse and reliable sperm supply. For example, females of Cryptopsaurus coesii have been found with up to 8 males attached. Biological and physiological mechanisms. The fusion process involves complex physiological changes, many of which are not fully understood due to the difficulty of studying deep-sea organisms. However, several key mechanisms have been identified. Immune system suppression. In most vertebrates, tissue fusion between the individuals would trigger an immune response, leading to rejection of foreign tissue. Anglerfish appear to have evolved a suppressed or modified immune response that allows the male's tissues to integrate with the female's without rejection. This is similar to mechanisms seen in organ transplantation or placental mammals, where immune tolerance is critical. Hormonal coordination. Hormones likely play a role in coordinating the fusion process and regulating sperm release. The female's reproductive hormones may signal the male's testes to produce and release sperm at the appropriate time. Nutrient dependency. Once fused, the male relies entirely on the female for nutrients as his digestive system degenerates. This parasitic relationship reduces the male's energy demands and allows the female to support multiple males without significant energetic cost. Evolutionary advantages. Sexual parasitism in anglerfish is a remarkable adaptation to the deep-sea environment, offering several evolutionary advantages. Guaranteed reproduction. In an environment where mates are difficult to find, the permanent attachment of males ensures that females always have access to sperm, increasing reproductive success. Energy efficiency. Males do not need to expend energy searching for multiple mates or surviving independently, allowing them to focus solely on reproduction. Adaptation to scarcity. The deep sea is a resource-poor environment, and the small size of males reduces their metabolic demands, while the female's larger size allows her to support multiple males. Genetic diversity. In species where multiple males attach to a single female, the female can use sperm from different males, potentially increasing genetic diversity in her offspring. Variations across species. Not all anglerfish species exhibit full sexual parasitism. The degree of fusion and parasitism varies. Permanent fusion. In species like Serratus caruncular and Cryptopsaurus coesii, the male becomes a lifelong parasitic appendage, fully integrated into the female's body. Temporary attachment. In some species, such as those in the family Linophrenidae, males may attach temporarily and retain some independence, detaching after sperm delivery. Non-parasitic males. In less extreme cases, some anglerfish species have free-living males that do not fuse with females, but instead release sperm externally. These variations reflect the diverse evolutionary pressures within the Ceratioideae suborder, shaped by differences in habitat, population density, and reproductive strategies. Challenges and mysteries. While the process of sexual parasitism is well documented, several aspects remain poorly understood. Immune mechanisms. The exact mechanisms by which anglerfish suppress immune rejection during fusion are still under investigation. Recent studies suggest similarities to immune tolerance in other organisms, but deep-sea research is challenging. Genetic basis. The genetic adaptations that enable this extreme form of parasitism are not fully mapped. as deep-sea anglerfish are difficult to study in captivity. Ecological impacts. The impact of sexual parasitism on population dynamics and genetic diversity in anglerfish populations is an area of ongoing research.